Biomolecules Part III. The living state. At this level, you must understand that in a living organism, tens and thousands of chemical compounds, otherwise called biomolecules or metabolites, are present at concentrations characteristic of each of them. For example, in a normal healthy individual, the blood concentration of glucose is 4.5 to 5.0 mm, while that of hormones would be nanograms per ml. In biological systems, the most important fact is that in a steady state exists all living organisms and are categorized by concentrations of each of these biomolecules. The biomolecules are present in a metabolic flux. At equilibrium, any chemical or physical process moves spontaneously. A non-equilibrium state is the steady state. From physics, one should understand that at equilibrium, the systems cannot perform work. The living organisms cannot afford to reach equilibrium as they work continuously. Hence, to be able to perform work, the living state is a non-equilibrium steady state. To prevent falling into equilibrium, living process is a constant effort. Energy input is achieved by this. For the production of energy, metabolism provides a mechanism. Hence, the metabolism and living state are synonymous. There cannot be a living state without metabolism. Enzymes. Almost all enzymes are proteins. Some nucleic acids are there that behave like enzymes, and these are called ribozymes. By a line diagram, an enzyme can be depicted. Like any protein, an enzyme has a primary structure, that is, amino acid sequence of the protein. An enzyme has the secondary and the tertiary structure like any protein. You will notice that when you look at a tertiary structure, the backbone of the protein chain folds upon itself. The chain crisscrosses itself, due to which many crevices or pockets are made. The active site is one such pocket. A crevice or a pocket is an active site of an enzyme into which the substrate fits. Thus, through their active site, the enzymes catalyze reactions at a high rate. In many ways, enzyme catalysts differ from inorganic catalysts, but one needs to mention a major difference. At high temperature and pressure, inorganic catalysts work efficiently, while at high temperatures, say above 40 degrees centigrade, the enzymes get damaged. However, from organisms who normally live under extremely high temperatures, for example hot vents and sulfur springs, the enzymes were isolated and were found to be stable, and even at high temperatures up to 80 degrees to 90 degrees centigrade, they retain their catalytic power. Thus, an important quality of such enzymes isolated from thermophilic organisms is thermal stability. Chemical reactions. Two types of changes the chemical compounds undergo. A change in shape without breaking of bonds simply refers to a physical change, and this is a physical process. A change in state of matter is another physical process. Water forms when ice melts or when water is converted into vapor. These are physical processes. However, during transformation, when bonds are broken, the new bonds are formed. This will be called a chemical reaction. Similarly, an organic chemical reaction is hydrolysis of starch into glucose. The amount of a product formed per unit time refers to the rate of a physical or a chemical process. If the direction is specified, rate can also be called velocity. Among other factors, rates of physical and chemical processes are influenced by temperature. in either direction for every 10 degrees centigrade change. A general rule of thumb is that the rate doubles or decreases by half at rates vastly higher than that of the uncatalyzed ones proceed the catalyzed reaction. When we observe the enzyme catalyzed reactions, the rate would be vastly higher than the same but uncatalyzed reaction. This reaction is very slow in the absence of any enzyme, with about 200 molecules of carbonic acid or H2CO3 being formed in an hour. However, by using carbonic anhydrase, an enzyme present within the cytoplasm, the reaction speeds dramatically with about 6 lakh molecules being formed every second. The reaction rate is accelerated by about 10 million times by the enzyme. Incredible indeed is the power of enzymes. Thousands of types of enzymes are present, each catalyzing a unique chemical or metabolic reaction. In a multi-step chemical reaction, when the same enzyme complex is or different enzymes catalyze each of the steps, it is called a metabolic pathway. For example, the chemical reaction is actually a metabolic pathway in which, through 10 different enzyme catalyzed metabolic reactions, glucose becomes pyruvic acid. At this stage, you should know that with one or two additional reactions, this very metabolic pathway gives rise to a variety of metabolic end products. Under anaerobic conditions in our skeletal muscle, lactic acid is formed. Pyruvic acid is formed under normal aerobic conditions. During fermentation in yeast, the same pathway leads to the production of ethanol. Hence, different products are possible in different conditions. How do enzymes bring about such high rates of chemical conversion? We have already understood the idea of an active site. A reaction is a chemical or a metabolic conversion. A substrate is a chemical which is converted into a product. Hence, enzymes, that is, proteins with an active site and three-dimensional structures, convert a substrate into a product. Symbolically, this can be depicted as S to P. It is now understood that within a given cleft or pocket, the substrate S has to bind the enzyme at its active site. The substrate towards the active site has to diffuse. There is thus an obligatory formation of an ES complex. E stands for enzyme. This formation of complex is a transient phenomenon. During the state where to the enzyme active site the substrate is bound, a new structure of the substrate called transition state structure is formed. The product is released from the active site very soon after the expected bond breaking or making is completed. In other words, the structure of products gets transformed from the structure of substrate. This transformation pathway of must go through the transition state structure. Between the stable substrate and the product, there could be many more altered structural states. All other intermediate structural states are unstable. Energy status of a molecule or a structure is related to stability. Hence, when we look at this pictorially through a graph, the potential energy content represents the y-axis and the progression of the structural transformation or states through the transition state represents the x-axis. You would notice two things between S and P: the energy level difference. The reaction is an exothermic reaction if P is at a lower level than S. In order to form the product, one need not supply energy by heating. However, the S has to go through a much higher energy state or transition state, whether it is an exothermic or spontaneous reaction or an endothermic or energy requiring reaction. The difference in this transition state from that of average energy content of S is called activation energy. Enzymes eventually make the transition of S to P more easy by bringing down this energy barrier. Nature of enzyme action. A substrate binding site in its molecule is present in each enzyme so that a highly reactive enzyme substrate complex is produced. This complex dissociates into its product P and is short-lived and the unchanged enzyme with an intermediate formation of the enzyme product complex. For catalysis, the formation of the ES complex is essential. In the following steps, the catalytic cycle of an enzyme action can be described. 1. First, to the active site of the enzyme, the substrate binds fitting into the active site. 2. When the substrate binds, it induces the enzyme to alter its shape, fitting more tightly around the substrate. A new enzyme product complex is formed when the active site of the enzyme, now in close proximity of the substrate, breaks the chemical bond of the substrate. 4. The products of the reaction are released by the enzyme and the free enzyme is ready to bind to another molecule of the substrate and once again run through the catalytic cycle. Factors affecting enzyme activity. By a change in the conditions, the activity of an enzyme can be affected, which can alter the tertiary structure of the protein. These include pH, temperature, binding of specific chemicals that regulate its activity, or change in substrate concentration. Temperature and pH. Generally, in a narrow range of temperature and pH, the enzymes function. At a particular temperature and pH, each enzyme shows the highest activity called the optimum temperature and optimum pH. Both below and above the optimum value, activity declines. In a temporarily inactive state, low temperature preserves the enzyme, whereas enzymatic activity is destroyed at high temperature because by heating proteins are denatured. Concentration of substrate. With the increase in substrate concentration, the velocity of the enzymatic reaction rises at first. Ultimately, a maximum velocity is reached by the reaction which is not exceeded by any further rise in concentration of the substrate. This is because the substrate molecules are more than the enzyme molecules, and after these molecules are saturated, there are no free enzyme molecules to bind with the additional substrate molecules. Also sensitive is the activity of an enzyme to the presence of specific chemicals that bind to the enzyme. The process is called inhibition when the binding of the chemical shuts off enzyme activity, and the chemical is called an inhibitor. In its molecular structure, when the inhibitor closely resembles the substrate and the enzyme activity is inhibited, it is known as competitive inhibitor. With the substrate, due to its close structural similarity, the inhibitor competes with the substrate for the substrate binding site of the enzyme. The substrate consequently cannot bind, and as a result the enzyme action declines. Example: malonate inhibits succinic dehydrogenase, which closely resembles the substrate succinate in structure. In the control of bacterial pathogens, such competitive inhibitors are often used. Classification and nomenclature of enzymes. Thousands of enzymes have been discovered, isolated, and studied. Based on the type of reactions they catalyze, most of these enzymes have been classified into different groups. Into 6 classes, the enzymes are divided, each with 4 to 13 subclasses, and by a 4-digit number, they are named accordingly. Oxidoreductases or dehydrogenases. Between two substrate S and S' enzymes which catalyze oxidoreduction. Transferases. Between a pair of substrate S and S', enzymes catalyzing a transfer of a group G. Hydrolases. Enzymes which catalyze hydrolysis of ether, ester, glycosidic, peptide, C-halide, CC or PN bonds. Lyases. Enzymes catalyzing removal of group from substrates by mechanism leaving double bonds other than hydrolysis. Isomerases includes all enzymes that catalyze interconversion of optical, geometric or positional isomers. Ligases, enzymes which catalyze the linking together of two compounds. Example, enzymes catalyzing joining of CO, CS, CN, PO, etc. bonds. Cofactors. One or several polypeptide chains are present in enzymes. However, a number of cases are there in which cofactors, the non-protein constituents, are bound to the enzyme to make the enzyme catalytically active. The protein portion of the enzymes in these instances is called the apoenzyme. Cofactors of three kinds may be identified: prosthetic groups, coenzymes, and metal ions. Prosthetic groups are organic compounds, and from other cofactors they are distinguished in that they are tightly bound to the apoenzyme. For example, in case of peroxidase, the catalase which catalyzes the hydrogen peroxide break down to water and oxygen. The prosthetic group is heme and is a part of the active site of the enzyme. Coenzymes are also organic compounds, but with the apoenzyme, their association is only transient, occurring usually during the course of catalysis. Furthermore, in a number of different enzyme catalyzed reactions, coenzymes serve as cofactors. Vitamins are the essential chemical components of many coenzymes. Example, vitamin niacin is present in coenzyme nicotinamide adenine dinucleotide or NAD and NADP. For their activity, metal ions are required by a number of enzymes which at the active site form coordination bonds with side chains, and at the same time one or more coordination bonds with the substrate are formed. Example, for the proteolytic enzyme carboxypeptidase, zinc is a cofactor. When the cofactor is removed from the enzyme, catalytic activity is lost, which testifies that in the catalytic activity of the enzyme they play a crucial role. Please share this video with your friends. Leave your comments below and please subscribe to my channel for more content. Click that bell notification that would let you know when a new video is uploaded. Thank you so much for watching. Once again, please subscribe to my channel for more content.